Okay, today's video is on glycolysis. So first we're going to have a quick overview of respiration. So respiration is a chemical reaction carried out to release energy. And there are two types of respiration, and one of them is aerobic respiration and the other is anaerobic. And here is a quick table summarizing the differences. So aerobic respiration is in the presence of oxygen. Anaerobic respiration is in the absence of oxygen. So aerobic respiration in the presence of oxygen is the general equation of glucose plus oxygen is carbon dioxide plus water. Whereas anaerobic respiration can happen in two ways. So there's the fermentation that happens in mammals. So this is glucose to lactic acid plus carbon dioxide. So this is what happens in our muscles when we don't have enough oxygen. And then there is the fermentation of glucose for carbohydrate crops. And for example, when making ethanol in industrial processes. So this is glucose to ethanol plus carbon dioxide. So this can be a way to produce ethanol, which can be used in fuels, for example. So aerobic respiration is more efficient at releasing energy, so it releases more energy per mole of glucose than anaerobic respiration, which is much less efficient at releasing this energy. So then we have to look at coenzymes before we go into the process of glycolysis. So an important fact is that coenzymes are not enzymes at all. Coenzymes are molecules that some enzymes require in order to function. So this diagram at the side shows one example of this. So this example is showing that the active site of the enzyme is not complementary to our substrate. It is an inactive enzyme. And it's this coenzyme that binds to this inactive enzyme that allows the substrate to be able to bind and activate the enzyme. Another example of this is coenzymes that carry hydrogen atoms from one molecule to another, and we'll see that in the process of glycolysis. So the important coenzymes that you will have to remember is NAD, and so the NAD is used throughout respiration and will be the coenzyme we're looking at in today's process. FAD, and this is important in the Krebs cycle, and the Krebs cycle is a later stage of respiration. And then there is NADP, and this is seen throughout photosynthesis. So these are the three important coenzymes used in chemical reactions that we will be studying. And NAD, which we'll be looking at in more detail today in the glycolysis process, works with dehydrogenase enzymes. So dehydrogenase enzymes are enzymes that catalyze the removal of hydrogen atoms from substrates. So these are enzymes that remove hydrogen atoms and they transfer them to other molecules involved in oxidative phosphorylation. And oxidative phosphorylation is the last stage of respiration, so we'll be looking at that later. But NAD is important because it works with these dehydrogenase enzymes to remove hydrogen atoms in order to use in later stages of respiration. So then before we go into these stages, we just need a quick recap of reduction and oxidation. So reduction is the gain of electrons, oxygen or hydrogen. Oxidation is the loss of electrons, oxygen or hydrogen. So it could be either of these depending on what process you're looking at. So coenzymes are reduced throughout respiration in this process. And so this could be FAD plus electrons, oxygen or hydrogen to get reduced FAD. And reduced FAD can also be written as this molecule here. Then there's NAD plus electrons, oxygen or hydrogen, depending on what part of the process we're looking at. And this makes reduced NAD. And, this should be N, but that makes NADH when you see it written down as a molecule. So now finally getting into glycolysis. So glycolysis is the first stage of respiration, and there are 4 stages of respiration. but glycolysis is the first stage. So glycolysis is a universal feature of every living organism. So every living organism will carry out glycolysis. And this can be evidence for evolution. So if every single living organism has this stage, it means that we could have evolved from all of these organisms at one point. So if we all share it, it means we must have evolved from each other. And glycolysis also takes place in the cytoplasm. So this means that this reaction doesn't require any organelles or membranes to carry out. So glycolysis has 4 stages. So in this first stage of respiration, there are 4 stages. And these 4 stages are the phosphorylation of glucose, the splitting of phosphorylated glucose, oxidation of triose phosphate, and ATP production. But we'll be looking into these 4 stages in more detail now. So the first stage of glycolysis is the phosphorylation of glucose. And so this box outlines where this process is happening. So this entire diagram is glycolysis, but the first stage of the phosphorylation of glucose is in this box. So we see that we start with a molecule of glucose up here, and this is 6 carbon atoms long. But we then have to look at the product at the end of glycolysis. So we look down here and we have these 3 carbon atom products. And as we start with a 6 carbon atom glucose, this means that we have to be able to split this glucose in 2, because we're ending with 2 3 carbon molecules. So we have to be able to split this glucose. However, we need to activate the glucose to make it more reactive, because we cannot just simply split this glucose. It is not reactive enough to be able to split. So we're going to need to activate this glucose to make it more reactive, so we're able to split it into these 2 3 carbon molecules to eventually get our product. So it is the addition of phosphate groups that activates our glucose. So activates means get more reactive. So it's the addition of phosphate groups that activates our glucose and lowers the activation energy of the coming enzyme-controlled reactions. So if we want to activate our glucose so we can split it, we need the hydrolysis of ATP. So hydrolysis is the breaking of bonds via the addition of water. So we want to hydrolyze 2 ATP molecules. As we can see here, our 2 ATP molecules are being broken down into ADP. So 2 ADP molecules are produced and 2 phosphates are produced. So these phosphates will go to bond with our glucose molecule to form our phosphorylated glucose. So now we have our phosphorylated glucose by the addition of our 2 phosphates from the hydrolysis of ATP. So this ATP that we have used provides the phosphates for the activation of our glucose, and it also provides the energy for this phosphorylation. So we have now activated our glucose successfully. So it's now phosphorylated. So now moving on to the second stage of glycolysis. So this is the splitting of phosphorylated glucose, because as we said before, we need to have these 2 3 carbon molecules, yet we still only have 1 6 carbon molecule. So we're going to need to split this. So now our phosphorylated glucose molecule is reactive enough and has a low enough activation energy to split. So we have successfully activated our glucose, so we can now split it. So the phosphorylated glucose is split into 2 triose phosphate molecules. So these are these molecules here. They have 3 carbons and 1 phosphate. So these are TP molecules, our triose phosphate. And just as a quick reminder, these triose phosphate molecules are the same molecules formed during the Calvin cycle in photosynthesis's light-independent reactions. So we can see the links between respiration and photosynthesis here. So in the second stage of glycolysis, we're taking our phosphorylated glucose and splitting it into 2 triose phosphate molecules. So now moving on to the third stage of glycolysis. So this is the oxidation of triose phosphate. So a quick recap again, oxidation is the loss of hydrogen. So it could be, for oxidation in general, loss of electrons, oxygen or hydrogen. But in this case, in the oxidation of triose phosphate, it's the loss of hydrogen. So each of our triose phosphate molecules that we've now formed from the splitting of phosphorylated glucose, so each of these triose phosphate molecules loses a hydrogen. So they are oxidized, the triose phosphate loses a hydrogen. And this hydrogen lost binds to NAD. So NAD is the coenzyme we were looking at in the beginning. So a coenzyme is a molecule that some enzymes require to function. So this NAD picks up this hydrogen and becomes reduced NAD. So NAD is our coenzyme, and it works as a coenzyme by being a hydrogen carrier molecule. So our triose phosphate is oxidized by losing a hydrogen, and our NAD picks up this hydrogen. It's a hydrogen carrier molecule, and by picking up this hydrogen, it becomes reduced NAD. And our last stage of glycolysis is ATP production. So in an enzyme-controlled reaction, triose phosphate is converted to pyruvate. So our oxidized triose phosphate that we have now produced via a chain of enzyme-controlled reactions is converted into our overall product of pyruvate. So it is important to watch out that there are steps missed out. So this green arrow actually represents many more reactions that are happening, but for the purpose of the A level, it's simplified into these 4 stages. So it may not all make complete sense when you put it together where some things come from or where they go, because there are reactions missed out for the A level for the purposes of teaching it in this way. So it's important just to accept that this is what happens, and don't get too confused about it because there are stages missed out. So during this conversion of triose phosphate into our pyruvate product, 4 molecules of ATP are produced, and this is by the phosphorylation of ADP. So 4 ADP molecules combine with a phosphate to produce 4 ATP molecules. So we have ATP produced during this enzyme control reaction of triose phosphate, oxidized triose phosphate because it's lost hydrogens, into our overall pyruvate. So now we've looked at the 4 stages of glycolysis. We're going to look at the overall outputs and inputs. So our inputs start with 1 glucose molecule. So we've started with 1 glucose, 1 6-carbon glucose at the beginning. And then we have 2 ATP molecules inserted into our reaction, and these are used up here to activate our glucose. So our ATP is hydrolyzed into our ADP and phosphate, so these phosphates can be used to activate our glucose into phosphorylated glucose. So we've used 2 ATP molecules. We've also used 4 ADP molecules, and this is used down here to form our ATP. So these ADP molecules are being phosphorylated to form our 4 ATP molecules. So we've used 4 ADP. And we've also used 2 NAD. So these are our coenzyme hydrogen carrier molecules. So our NAD are here. They've been inserted into the reaction to carry these hydrogen molecules. So we've used 2 NAD. And our overall outputs are 2 pyruvate molecules. So these are down here, our overall product. And we've also used 2 ADP. We've produced 2 ADP. So during our activation of glucose, our ATP has been hydrolyzed into ADP and phosphate. So our phosphates have been used to activate and phosphorylate our glucose, whereas the 2 ADP produced are not used, so they are our overall output. And we've also produced 4 ATP. So during the phosphorylation of ADP, during our enzyme-controlled reaction of TP to pyruvate, we have produced 4 ATP molecules. And we've also produced 2 reduced NAD. So during our oxidation of triose phosphate, our triose phosphate has lost hydrogens, and this has gone to reduce our NAD that is now reduced NAD, and these are used later on in respiration. So those are our 4 stages of glycolysis. There's the phosphorylation of glucose via ATP, and this is to activate our glucose so we can split it. So our phosphorylated glucose is then split into 2 triose phosphate molecules. Our 2 triose phosphate molecules are oxidized by losing hydrogens, which are picked up by our NAD, and this produces reduced NAD. And so then our triose phosphate molecules can go on to be converted into pyruvate via enzyme-controlled reactions. And during these enzyme-controlled reactions, we produce 4 ATP molecules via the phosphorylation of ADP.